welcome to another deep dive. This time, into the fascinating world of viscous fingering. We're going to try and answer the question, is it totally chaotic and unpredictable? Or can we find some patterns?
Interesting question, yeah.
To help us figure that out, we're diving into a 2007 paper. It's called Reproducibility of the Finger Pattern in Viscous Fingering.
Okay.
And it's published in the Journal of Chromatography.
Yay. That's good.
The authors are Shalliker, Victor Wong, and Georges Guiochon.
Now, before we jump into the paper itself, let's make sure everyone's on the same page. Can you give us a quick rundown of what viscous fingering is?
Sure. So you can picture it like this. Imagine a porous medium like you'd find in a chromatography column.
Okay, yeah.
And you have one liquid pushing through another liquid that's more viscous, thicker. The less viscous liquid, it wants to move faster, right? So it kind of forms these finger-like projections as it displaces the thicker one. And that creates these kind of cool patterns. They're called viscous fingering. And this isn't just some theoretical thing, right? It has real-world implications.
Oh, yeah, definitely. Viscous fingering, it pops up in a bunch of different fields. It's a big deal in chromatography, for example. Yeah. It can really mess with how well components separate in a mixture.
Right. Which makes sense if you think about how those fingers are mixing things up.
Exactly. So if you're trying to optimize the separation, you really need to understand and control this phenomenon.
And that brings us back to the paper.
Yeah.
The big question Shalliker, Wong, and Guiochon are trying to answer is, are these fingering patterns totally random? Or is there some rhyme or reason to how they form? And to do this, they came up with a really clever way to actually see this happening in 3D inside a chromatography column.
Right. It's really cool.
I know scientists initially used 2D models to study this, like Hele-Shaw cells. Why weren't those enough?
Well, Hele-Shaw cells, they're like these thin, flat chambers. They're useful for basic observations, but they don't really capture the full complexity of what's going on in a real column. It's a 3D thing.
So they needed to find a way to see inside that 3D space, which is where their breakthrough technique comes in. How they managed to actually see viscous fingering in 3D.
Well, basically, they created a chromatography column that was, get this, optically transparent.
What? How do you even...
It's pretty ingenious. They matched the refractive index of the liquids and the packing material inside the column so precisely that it basically disappeared.
They made a whole column invisible. That's wild.
It is. And then they added a dye to the less viscous liquid so they could track how it moved and displace the other liquid.
So they could actually watch those finger-like projections form in 3D in real time.
Yep. It was something that had never been done before.
Amazing. So what were some of the first things they saw with this setup? Did they start to find any patterns?
One of the most surprising findings, at least initially, was that under certain flow conditions, the fingering patterns were actually quite reproducible. Like when they kept the flow really slow, they'd see almost the exact same finger patterns forming over multiple experiments.
So slow and steady does seem to have its advantages, at least in the world of viscous fingering.
Yeah, who knew? But of course, there's always a but.
Of course.
It turned out that things got a lot more complicated as they started to tweak different factors, like the flow rate, the difference in viscosity, even how the sample was distributed at the top of the column.
Sounds like there's a lot more to unpack here. We'll have to dive into those details and the implications of their findings after a short break.
Yeah, so to really understand what they did, we need to look closer at how they actually created this invisible column.
Okay, yeah, let's break that down a bit. Just sounds pretty amazing. How they managed to make the column transparent.
So basically, it comes down to refractive indices. They carefully chose liquids and packing materials that had, almost identical refractive indices. By matching them so closely, they basically eliminated the scattering of light that would normally happen as it passes through the column. So it just vanishes.
It's like they gave it a perfect optical camouflage.
Exactly. And that transparency, that was key because it let them use a dye to visualize how the less viscous liquid moved and displaced the more viscous one inside.
Right. Without that transparency, they wouldn't be able to actually track those finger patterns in 3D. So they made the column invisible, they added the dye. What kind of equipment did they use to actually capture all this?
Well, they used both film photography, like a 35mm camera with a macro lens to get those close-ups, and they also used a digital video recorder.
So they were getting both high-resolution snapshots and video of the whole process.
Yep, the best of both worlds.
Yep.
And they didn't stop there. They took those film images, digitized them, and used some pretty sophisticated software to analyze those patterns, get some real data out of them.
And that makes sense. It's one thing to see it, but to really understand it, you need to quantify it, right?
Exactly. And they were really meticulous about it. Like they'd overlay grids on the images to pinpoint the exact location of each fingertip.
Wow, so they were literally mapping out the coordinates of those viscous fingers.
Yeah, pretty cool, huh? That way, they could compare the positions of those key fingers across different runs of the experiment and really see how reproducible the patterns were.
So they could see if those fingers were forming in the same spots each time. Did they use any other techniques to compare the pattern?
Oh, yeah, they also did this thing called digital image subtraction. Basically, they'd take images from two different runs and subtract one from the other.
Interesting.
So if the patterns were totally identical, the resulting image would be completely white. But any differences between the patterns would show up as darker areas.
Ah, so it's like a visual representation of how similar or different the patterns were. Clever.
It is. It really highlights those subtle variations you might miss with just a side-by-side comparison. And, speaking of things that can affect the patterns, we talked earlier about how the initial distribution of the sample at the top of the column can matter.
Right, you said an uneven distribution could throw things off.
Yeah.
Can you explain why that is?
Well, think about it. You introduce the sample at the top of the column, right? Ideally, you want that sample to spread out evenly across the whole width of the column before it starts moving down.
So it's like a level playing field.
Exactly. But if the distribution's uneven, it can create variations in the flow pattern right from the start, and that can make the viscous fingering even more unstable.
It's like starting a race with some runners way ahead of the others. It's not going to be a fair competition.
Exactly. And to study this, they actually used two different column designs. One that promoted a more even sample distribution at the inlet, and one that led to a less even distribution.
So they could see how that initial setup affected things downstream.
Right. They wanted to isolate that variable and see how it impacted the reproducibility of the patterns. And what they found was, unsurprisingly, a more uniform sample distribution led to more consistent fingering patterns.
It seems like they really thought of everything. It's impressive how carefully they controlled every aspect of the experiment.
Yeah, they were very thorough. And because of that, they were able to make some pretty groundbreaking discoveries. Now, are you ready to dig into those findings and see what they actually learned about the predictability of viscous fingering?
Absolutely. Let's get to those results.
There.
Okay, so let's dig into those results. What did they actually find out about, whether viscous fingering is predictable or not?
So as we talked about earlier, at those really low flow rates, they saw a lot of reproducibility, like almost identical patterns forming multiple times.
Right, so slow and steady wins the reproducibility race.
Yeah, exactly. And that actually challenged what a lot of people thought about viscous fingering. They thought it was always chaotic, but this showed that if you control things just right, you can get some order out of it.
But I bet it didn't stay that simple. Right, they must have tried changing other things.
Of course. As soon as they started increasing the flow rate, they saw less and less reproducibility, faster flow, more unpredictable patterns.
Well, yeah, I guess the faster things are moving, the more turbulence there is. It's got to be harder to get things to behave the same way every time. Right.
Exactly. So it's not just about finding a specific flow rate. It's about finding that sweet spot where things are moving fast enough, but still staying consistent.
So flow rate is definitely a big one. What about the liquids themselves? Like, how much of a difference there was in viscosity? Did that matter?
It did, yeah. They found that if the difference in viscosity between the two liquids was smaller, the patterns were more reproducible.
So a gentler kind of transition between them.
Exactly. It's like that contrast in viscosity is what really drives the instability. So a smaller contrast seems to make things a bit smoother, more predictable.
So we've got flow rate, viscosity difference, and then there's that whole thing about how the sample is distributed at the top of the column.
Oh yeah, that's a big one too. Remember how we talked about how an uneven distribution can really mess things up?
Yep, gotta have that level playing field.
Right. And their experiments really proved that when they started with a more even distribution, they got much more consistent patterns. It all comes back to that idea of control, really.
It's pretty incredible when you think about it. They took this phenomenon that seems so random and chaotic and started to tease apart all these different factors that influence it.
Yeah, and their work has some really important implications for other fields too, like in chromatography, where you need those really precise, repeatable separations.
Right. If you're trying to analyze something, you need to be able to trust that your results are going to be the same every time.
Exactly. And this kind of research helps us understand how to get those reliable results. It's like, by understanding how viscous fingering works, we can design better separation processes, maybe even improve the accuracy of all kinds of analytical techniques.
It's like having a new set of tools to fine-tune things. And it's not just chromatography, right? This could be useful anywhere you've got fluids flowing through porous media.
Oh, yeah, for sure. Think about things like oil recovery, even understanding how groundwater moves through soil, all sorts of applications.
I've got to say, this has been a fascinating deep dive. It's really amazing to see how something that seems so abstract can have such wide-reaching implications.
It is, isn't it? It just goes to show you, sometimes the most unexpected discoveries can lead to the most valuable insights.
Thanks for taking this deep dive with us. Hopefully it's given everyone some food for thought, maybe even sparked some new ideas for harnessing the power of viscous fingering.